effect of tonicity on erythrocytes.
Secondly, to find out the effect of NaCl concentration on the percentage of hemolysis of erythrocytes.
And finally, to differentiate between tonicity and osmolarity.
In section A1, label the centrifuge tube as such: hypertonic, hypotonic, isotonic and control, which is the distilled water. Add 5 ml of sodium chloride into each test tube. Then, add 50 microliters of blood into each test tube by using micropipette. Next, cover each test tube with paraffin and mix the content gently. And leave the tube at room temperature for 20 minutes. After 20 minutes, carefully mix the content again and centrifuge all tube for 5 minutes at 200 rpm in the centrifuge machine. Carefully transfer all the supernatant into cuvette by using dropper. Determine the percentage of hemolysis in each tube by measuring the absorbency of the supernatants. Set the spectrometer at a wavelength of 540 nanometer and zero it using the blank. Then, measure the absorbency of each supernatant with the same wavelength. Compare the absorbency of the test supernatants to that of the supernatant from the cells treated with distilled water. In section A2, repeat all the experiment with the same procedure as section A1 by using this sodium chloride solution with different percentage. Plot a graph for the percentage of hemolysis versus sodium chloride concentration. In the last section, section B, repeat all the experiment with the same procedure as section A1 and A2 by using the following solution. Lastly, calculate the percentage of hemolysis for the supernatants by using this equation. Percentage of hemolysis is equal to absorbency of supernatant divided by absorbency of supernatant from the tube treated with distilled water times 100.
In the first section, the erythrocytes are immersed in the hypertonic, hypotonic, and isotonic solution and distilled water as a control for the comparison in the next section of experiment, that is A2 and B. Hypertonic, hypotonic, and isotonic solutions are actually under tonicity. Tonicity is a measure of the osmotic pressure gradient of two solutions separated by a semi-permeable membrane. A hypertonic solution has a higher concentration of solutes than does the cytosol inside the erythrocytes, so the molecules move out of the cells faster than they enter, causing the cells to shrink. Such shrinkage is called crenation.
A hypertonic solution has a lower concentration of solute if we compare it with the cytosol of the erythrocyte. This will make the water molecules to enter the cell and will eventually cause the cell to swell and burst. Meanwhile, in an isotonic solution, the concentration of solute is equal with the cytosol. So, the water molecule will enter and leave the cell about the same rate. We will not find a significant change in the shape and volume of the cell.
In this experiment, we observed that the percentage of hemolysis in hypotonic solution is 57%, 0.59% in hypertonic solution, and 1.49% in isotonic solution. The percentage of hemolysis is relatively higher in hypotonic solution compared to isotonic and hypertonic solution. Besides, the color of the supernatant in hypotonic solution is red, while clear in both hypertonic and isotonic solution. The reddish color of the supernatant can be explained as the presence of red hemoglobin pigment in the erythrocyte. This pigment doesn't have mass. When in hypotonic solution, hemolysis occurred, causing the hemoglobin in the red blood cell to move from the cell to the solution.
In isotonic and hypertonic solution, clination occurs, meaning that the water molecules that moving out of the cell is faster than into the cell. So the cell shrink that cause the hemoglobin inside the cell to remain. Hence, after centrifuge, there is some red deposition in this solution because hemoglobin cannot separate from its side. Thus, it will trap in the cell. Plus, the cell has its own mass. That is why it will deposit on the bottom of the solution and the upper solution will remain clear. In distilled water, most of the hemolysis occurs here because this is the most hypotonic solution. So the hemolysis occurs 100% in this solution. It acts as a control in this experiment. Hence, the higher the concentration of sodium chloride, the lower the percentage of hemolysis.
In this section, the experiment is carried out using different concentration of sodium chloride, NaCl. NaCl is common salt of sodium that present in all tissues and important in maintaining the electrolyte balance of the body. From the graph plot, the percentage of hemolysis is decreasing with the increasing concentration of NaCl. There is high significant difference in percentage of hemolysis between 0.4% and 0.45%. It is observed that the NaCl concentration with 0.4%, 0.35%, 0.2%, 0.1% and distilled water are hypotonic to the erythrocyte because the percentage of hemolysis is high. This is due to low amount of non-penetrating solute in the solution compared within the cell and water molecule diffuse into the cell and causing hemolysis. Meanwhile, the rest concentration 0.45%, 0.50%, 0.55%, 0.65%, 0.75%, 0.85% and 1% are shown to be hypertonic because it have a low percentage of hemolysis. This might be due to the high amount of non-penetrating solute in the solution compared to the cell and water molecule diffuse out from the cell and causing shrinkage. In this experiment, the percentage of hemolysis for 300 milliosmol of sucrose, 300 milliosmol of glucose, and 300 milliosmol of NaCl are 3.33%, 5.12%, and 0.89% respectively. There is low percentage of hemolysis because the amount of water molecule leave and enter the cell are the same rate. Thus, no clination or hemolysis occurred that made the supernatant remain clear without any hemoglobin inside. As for 300 milliosmol, 150 milliosmol, and 600 milliosmol of urea, the percentage of hemolysis are 93.58%, 93.41%, and 97.32% respectively. The high percentage of hemolysis of the red blood cell proven that the solution are hypotonic to cell. They are penetrating solute, which means that they cannot hold water. They enter the cell, make it swelling, and eventually burst. This will make the supernatant turn red in color. The percentage of hemolysis using 300 milliosmol of urea in 300 milliosmol of NaCl is 1.06%. Due to the presence of the penetrating solute, which is urea, there should be a high percentage of hemolysis, same as the other solution of urea. However, in the presence of non-penetrating solute and HCl, the rate diffusion of penetrating solute and water molecule in and out of the cell occur at the same rate. Thus, there is approximately no change to the red blood cell and no hemolysis of the hemoglobin occur.
There are some precautions that need to be taken during this experiment to avoid the mistakes on the result. First, balance the test tube in terms of equal volume and same side during centrifuge to avoid the solution from spill out. Secondly, throw the blood sample into the appropriate sink and wear the glove all the time to prevent contamination of virus and bacteria. And lastly, the blood was mixed gently with the solution to prevent the rupture of the erythrocyte because erythrocyte is quite fragile.
In a conclusion, the hypertonic and isotonic solution will appear as a clear supernatant, while the hypotonic solution will appear as a red supernatant.
The hypotonic solution will cause water to move inside the erythrocyte and then cause the cell to swell. Thus, the solution will turn reddish in color.
The hypotonic solution will cause the water to move outside the cell and shrinkage of the cell will occur. Thus, the supernatant will appear as clear and have a few agglutination of blood at the bottom of the test tube.
From this experiment, I can find out the effect of tonicity on erythrocytes.
Next, we are also able to learn the use of technology. For example, the use of centrifuge machine.
We managed to find out the effect of sodium chloride concentration on the percentage of hemolysis, and we successfully plot the table and graph.
Besides, we are able to differentiate between tonicity and osmolarity. Thus, we learn the meaning of these two terms.
Finally, I have learned that everything that occurs in our body has its own reason. So be grateful for that. Remember, take care of your health so that you can have a better life. So I think that's all for now. Bye!